An introduction to why we're doing this. So I started my SciDev journey many lifetimes ago because I wanted to help people. I wanted to use my interest in science and mathematics to understand diseases and common afflictions and health problems for us, for all of us, for humans, and try to understand that and therefore try to develop treatments and therapeutics to improve healthcare and human well-being in general. And that aligns perfectly with Rosalind as a company. This mission, it aligns all of us and it drives us all towards making a really great platform that all of you can use to drive your innovation. So we're here to empower you in this global mission. But you unlock the true value and you get the synergy when you combine them together and you extend them from one end to the other. And I'll talk a little bit about this in more depth. This is broken down into 6 sessions. We covered 2 weeks ago, custom pruning of low expressors. Today, we're focused on custom normalization. In the Rosalind platform, there are different modules to analyze your data. So we'll be seeing some of these in more detail as we progress over the next 5 sessions. Today, we're still in the experimental setup stage. So we'll be looking at how to set up your experiment, how to normalize your data, how to do some nice customizations as well. And Rosalind and nCounter work together beautifully because it's a very fast technology to generate your data. You plug that directly into the Rosalind platform, and then you can start benefiting from all of the enhancements, the intelligence capabilities to extend the value of your targeted panel and get the most out of it that you can. Why are you performing normalization? But I want to challenge you to think about the basic principles of why we normalize, how we do it, and the choices we make, because the more you understand this, the better your analysis is going to be. It can be distilled in this short segment here. We want to adjust the raw data in preparation for downstream biological interpretation, such as differential expression analysis. And so we're adjusting for 2 different sources of variation. There's biological sources of variation, there's technical sources of variation. So let's talk about the normalization, how it's done, the choices we make, and why we do it. First step is we want to identify probes to use in normalization. And I listed some best practices here. Second step is that you want to then apply your normalization algorithm to the raw data to generate your normalized counts. But quickly, within a second or less, this is obviously raw data. The reason we know this is because if you understand the chemistry and how the technology works, is that it's single molecule counting. So these are integers, 279. So what this represents is 279 counts of the barcode for this ABCF1 gene in sample number 1. So in other words, 279 molecules or 279 transcripts. So on the left is the formula for standard deviation. And on the right is the formula for the coefficient of variation, or I should say the percent coefficient of variation. And at the top, I have my gene CC2D1B. It has the lowest standard deviation and a rank order of 1, meaning it has the lowest standard deviation. And if I scroll down, you'll see that the SDs increase as well as the rank order. So the lower the rank order here, the lower the standard deviation. And so this is just ordered from low to high. So important thing to note here is, the lowest or the most stable gene, 5% ZV, is rank order 1. But if we look at standard deviation, it's actually 7. Another thing that popped out to me is that this gene here, whatever it is, CC2D1B, when we look at its percent CV ranking, it's 26, so not very stable, but it's actually number 1 by standard deviation. So I think you might be starting to understand why standard deviation isn't a useful metric. And let me show you a couple figures to drive this home, kind of bring this to life here. You can normalize OGG1 by individual housekeeping genes, and you get a completely different result depending on the gene you normalize with after you upload your RCC files. And there's 2 paths. There's this default path and advanced path. The advanced workflow is available to our paid subscribers. So all of our academic researchers, our academic subscribers, professional enterprise subscribers can access the advanced path. If you have a sponsored subscription via NanoString, you have option to the default path. So if you do want to access the advanced paths, you will need to upgrade your subscription. So let's take a look at the advanced paths and the advantages of having this available. So this is custom pruning screen, which we talked about last time. I'm not going to go into depth. So historically, in other software packages, you have selected your housekeeping genes for normalization first, and then you've pruned the low expressors. What that can result in is that you can actually have some housekeeping genes below background or below your negative controls enter the normalization process, which clearly you want to avoid because you're just introducing noise into your normalization, and that just cascades downstream, adding noise to all of your interpretation. Yeah, thanks everyone for joining. I was really happy to see the turnout, and thanks for all the questions and the engagement. Definitely want to share my knowledge with all of you and help you be successful. Hope to see you in the next session coming up, quality control. Our attendance and registrations keep increasing over time, so this is really great to see. In the meantime, before May 16th, if you have questions, please reach out to our customer success and support teams. If not, we will see you on May 16th. Thank you.